Meiosis. Meiosis is divided into meiosis 1 and meiosis 2. And we're going to go on and start with what would be called a primary spermatocyte, for example. So in prophase 1, the chromosomes shorten and thicken and become visible with a compound light microscope. The centrioles are moving to the opposite poles. The spindle fibers are forming. But something different happens. And that is the homologs touch each other. And they swap pieces of DNA. Okay? So we call this crossing over. So we have this new thing that happens that does not happen in mitosis. Crossing over. So that then and here's how they line up in metaphase. It is different. I'm going to show my result of crossing over. They line up like this. So instead of all 4 of them lined up, the homologs are still paired. So that in anaphase 1, let me make this one shorter. What we have pulled apart are homologs instead of sister chromatids. So that when we get to telophase, then we may or may not have a nucleus reform, but I'll put one in there. Because this is kind of, some of these steps are abbreviated because in general these cells are going to jump right back in, or jump into meiosis 2, so that one cell gets this homolog, shorter, and the other gets this one. Or each for the long and the short. So the product here is haploid. It only has one from each pair. Now we still have sister chromatids connected, and those need to be separated. That's what meiosis 2 is all about. So it's different in that now we're following 2 cells. If the chromosomes lengthened, they shorten. And thicken. pain in the rear to draw the products. That didn't even work out for me. The products of crossing over. Okay, so we have 2 cells now going through meiosis 2. So prophase, metaphase, now the sister chromatids are lined up. Shorter. Okay. Anaphase, they're pulled apart. Okay, so that by telophase, getting our new nucleus, or nuclei, look at this. Woo. And we have oops, wrong color. Trying to hold too many of these. I'm trying to color the carpet. Oops, wrong color. The short one should be blue. On it. This should be more blue with a little bit of red. This one should have a little blue and a little red. Okay, and then they would divide into those cells with their double helices present. So you'll be asked to compare and contrast meiosis and mitosis, so I want you to think about that. Which one gives you the identical daughter cells that are also diploid? Which one gives you haploid cells, etc. All right, now I want you to look at this, and with the exception of a cell right here that's called a spermatogonian, think of spermatogenesis. So we've got this cell here. It is diploid. It is, in fact, it divides, and when it divides by mitosis and produces more spermatogonia. All this starts when a male hits puberty, and so he's got a whole bunch of spermatogonia, and they are, let's say that with every mitotic division, every mitosis that occurs, one of those spermatogonia stays a spermatogonian or a germ cell, and the other one differentiates into a primary spermatocyte. The primary spermatocyte undergoes meiosis 1. So this would be a primary spermatocyte. And the result of meiosis 1 would be 2 secondary spermatocytes. So these would be our secondary spermatocytes, then each of those would undergo meiosis II, and each of them would produce 2 immature sperm called spermatids. That's spermatogenesis. That's how it works. With oogenesis, it's different. While a female is a fetus, so she's still in her mom's uterus, she is a fetus. What happens is she has oogonia that are dividing by mitosis. Some of them differentiate into primary oocytes. And what they do is they do their crossing over, and then they go into metaphase and they stop. They stop. And the fetus continues developing. The baby girl is born. She's a baby, she's a toddler, she's a little girl, she's a big girl, and she hits puberty. Once she hits puberty, if she doesn't take any hormonal methods of birth control, and she doesn't get pregnant, once a month, one of these primary oocytes resumes meiosis I. So it resumes meiosis I, and something different happens here. What happens is one secondary oocyte is formed, and then this teeny tiny polar body. And in general, nothing happens with the polar body. It's done. So that instead of having 2 cells down here, there's one secondary oocyte. And that one starts meiosis II. It arrests again, and if fertilization does not occur, it is flushed from the body as a secondary oocyte. If it does, if fertilization occurs, it will, once that sperm gets through the cell membrane, boom, meiosis II finishes very quickly producing, and again, not 2 cells, 1 egg, 1 nucleus for the egg, and another polar body, boom, and the fertilization of the egg, and then that's when the zygote forms. Yeah, so oogenesis and spermatogenesis are different, and you need to be able to compare and contrast those as well. A lot of you are very interested in non-disjunction. It is an interesting topic. First, let me say, problems, deformities, or diseases that are genetic, there are different categories, so let's look at this. We have, or there can be, chromosomal abnormalities, which that's the topic of your discussion this week, and you have different chromosomal abnormalities that you all are looking into. So there are, I'm going to set this down so it doesn't fall on me. So there are chromosomal abnormalities. Those are not genetic disorders. They are issues with non-disjunction. That is the problem with non-disjunction. During meiosis, either homologs or sister chromatids do not separate properly, so that the resulting cells get too many or too few of a given chromosome. So it's not all 23 pairs, it's 1, and you're going to get to look into some of those. Everyone should get to see 3 of them, because they'll do their own, and they'll respond to 2 students, and they'll have different ones. So that'll give you 3 examples of non-disjunction to look at, or chromosomal abnormalities. Then there are genetic diseases, and that is where there's a mutant gene that has some change in the genetic code, The DNA is different, so the messenger RNA codons are different, so that the protein is different. That's our genetic disorders like cystic fibrosis and classic hemophilia. We'll talk about those next week. And then there are, somebody asked about intersex and all that. Those kind of things, hermaphroditism in humans, those things happen when there's a mistake that occurs during development. So that it's amazing. Cells, when a zygote forms, it's that fertilized egg. It divides into 2 cells, then 4 cells, then 8 cells, then 16, then 32. And it keeps dividing, and for a while all the cells are the same. But then the cells start to differentiate. And as they start to differentiate, they start to move around. And as they move around, if they end up in the wrong place at the wrong time, or they